Welcome to microbiology class. In today's class, we are going to see morphology and fine structure of bacteria. So bacteria, they are the largest group of microorganisms of medical significance. They are prokaryotic in nature. They are single-celled organisms. They lack nuclear membrane, metabolically active, and they divide by binary fission. Coming to the size of bacteria, the bacterial size, it ranges from 0.2 to 2 micrometer in diameter and 0.5 to 5 micrometer in length. Two largest bacteria example, Thiomargarita namibiensis, Eupulopsicum fischelsoni. They are visible through naked eye, and the smallest bacteria size ranges from 0.2 to 0.3 μm, example Mycoplasma. So Eupulopsicum fischelsoni, the size it ranges from 200 to 700 μm. Thiomargarita namibiensis, the size it ranges from 100 to 300 μm. Coming to the shapes of bacteria, commonly three shapes of bacteria are available. One is coccus, and the second one is rod shape, and the third one is spiral shape. Within the coccus, there are subtypes such as Diplococcus, Streptococcus, tetrad arrangement, Staphylococcus, Sarcine. And then coming to the rod-shaped bacteria, Diplobacillus, Streptobacillus, trichome arrangement, palisade arrangement, Coccobacillus, and then comma-shaped bacteria, Vibrio. And coming to the spiral-shaped bacteria, example Spirillum, Spirochete, and within the spirochete example, Treponema palladium, which causes syphilis. And then filamentous bacteria, box-shaped bacteria, appendaged bacteria, and some bacteria, they do not have exact shape. They are called as pleomorphic in nature, pleomorphic bacteria. So these are all the different shapes of bacteria. This is the overview or overall diagram of bacteria. So in this figure, we can see both the external structure of bacteria and then the internal components of bacteria. We will see in detail about the external structures one by one. So the first external structure is capsule. So this is the outermost covering of bacteria, and this is surrounded by polysaccharide. It binds to the cell surface, and this protects the bacteria from phagocytosis, that is engulfment of bacteria. And this protects the bacteria from macrophages and neutrophil attachment. They are firmly attached to the cell surface. Example for capsule containing bacteria, Streptococcus pneumoniae. And the next one is slime layer. This is also similar to capsule, but this is loosely attached to the cell surface. And this also binds to different cell surface. This evades the phagocytosis. And the next external structure is flagella. So this is the outermost thread-like structure which is present in bacteria, which is used for locomotion of bacteria from one place to another place. This flagella, it is made up of flagellin protein, and this helps in the movement of bacteria, chemotaxis. There are different types of flagellar arrangement. If a single flagellum, if it is present at one end, it is called as monotrichous. Small bunch of flagella arises from one end, that is called as amphitrichous. If the flagella are present at both the ends of bacteria, they are called as amphitrichous, and peritrichous means the flagella which is present all over the body of bacteria. So these are all the flagellar arrangement: monotrichous, lophotrichous, amphitrichous, and peritrichous. Then the next external structure is pili. So here we can see small projections which is present surrounding the bacteria. The pili, it is shorter than fimbriae. So the pili, it is hair-like microfibrils binding to the surface of the body cells. Example, mucosal cells. This pili, it involves in bacterial conjugation. So this is used for transferring the genetic material from one bacteria to other bacteria. This is present only in gram-negative bacteria. Highly containing organism, example, Shigella, Protease, Neisseria gonorrhoeae, E. coli, etc. And then the next external structure is fimbriae, tiny bristle-like fibrils attached to the substrate, and this fimbriae, it is present both in gram-positive and in gram-negative bacteria. Then the next important external structure is the cell wall. So the bacteria, they are classified into two types based on the cell wall composition. One is gram-positive bacteria and another one is gram-negative bacteria. Cell wall composition of gram-positive bacteria, it consists of peptidoglycan, lipid, and teichoic acid. And the cell wall composition of gram-negative bacteria, it consists of peptidoglycan and then one more outer membrane, lipid, protein, lipopolysaccharides are present. So in this diagram, we can easily differentiate between the gram-positive bacteria and gram-negative bacteria. From the structure of the cell wall, we can easily differentiate that thick peptidoglycan layer, it is present in gram-positive bacteria, and then thin peptidoglycan layer, it is present in gram-negative bacteria. Then the next important difference is outer membrane, it is present in gram-negative bacteria, and this is absent in gram-positive bacteria. Periplasmic space, it is very less in gram-positive bacteria and it is very thick in gram-negative bacteria. Plasma membrane and integral proteins are present both in gram-positive and in gram-negative bacteria. This outer membrane, it consists of lipopolysaccharides, porins, and then O-specific side chains are present in the gram-negative bacteria. Then the next important external structure is the plasma membrane. So this plasma membrane, in this first diagram we can see, here we have seen about the capsule and then we have seen about the cell wall. The next layer about the plasma membrane we are going to see. So they are semi-permeable in nature, selective permeability. Energy reactions takes place in the plasma membrane, synthesis of molecules, transport of solutes, energy generation, and location of numerous enzyme systems are present in the plasma membrane. They are made up of phospholipid bilayer. So the phospholipid, we can see hydrophilic head, which is present at the outside, and the hydrophobic tail portion, which is embedded inside the plasma membrane. So here, the structure of the plasma membrane, it consists of phospholipid bilayer and then integral proteins and peripheral proteins are present for allowing the transfer of ions, solutes to enter inside and go outside of the cell. Coming to the internal structure, first one is the cytoplasm. Cytoplasm, it contains water, enzymes, ribosomes, mesosomes, inclusion bodies, and cellular DNA. Coming to the important part of the internal structure, that is the nucleus. It is the most important part of the cell. It controls and directs all the cellular activities and stores the hereditary information of the cell. So the bacterial nucleus, it is known as nucleoid because it lacks nuclear membrane, nucleoplasm, and nucleolus is absent. Bacterial DNA, it is naked in nature. It lacks histone proteins. The function of the bacterial nucleoid is it contains and stores the hereditary information of the cell and it controls all the cell activities. And then next important internal structure is plasmid. It is also present in the cytoplasm. Independent circular DNA, additional genetic informations are present in the plasmid. They get transferred between the bacteria by conjugation process through binding. And then the next important internal organelle is Ribosome, the structures that have multiple components and they are responsible for protein synthesis. So the ribosome which is present in prokaryote is 70S ribosome, which consists of larger subunit 50S and then small subunit 30S. Other inclusion bodies are present, such as nutritional sources, glycogen granules, starch granules, beta hydroxybutyrate granules, gas vesicles are present. So in this diagram, you can see the inclusion bodies. And the next important internal structure is mesosome. Here in this diagram, you can see the mesosome. So this is spherical or round sac-like structure found commonly in gram-positive bacteria. So this is the site for respiration in bacterial cell. And then coming to spores. So spores are metabolically dormant structure produced during unfavorable condition by the process called sporulation. If there is no effective temperature, pH or nutrient source, the bacteria will survive by producing dormant spores. So this sporulation, it occurs during the late log phase or early stationary phase. There are different phases of bacterial growth. One is lag phase, log phase, stationary phase, and death phase. So this sporulation stage, it is attained during the late log phase or early stationary phase. Under favorable conditions, the spores germinate to give the vegetative cell. And example for spore-containing bacteria are Bacillus stereothermophilus, produces spores, which is used for quality control of heat sterilization equipment, such as for checking whether the autoclave is working properly or not. For that, we can use this Bacillus stereothermophilus spores. And Bacillus anthracis, this is one example for spore-containing bacteria, which is used in biological warfare. So we have seen in detail about the internal and external structures present in bacteria. Thank you.